Welcome to Going Antiviral, the podcast of the IAS-USA, a professional continuing medical education organization focused on HIV and other viral diseases. I'm Dr. Michael Saag, professor of medicine and infectious diseases at the University of Alabama at Birmingham and volunteer member of the IAS-USA Board of Directors. Welcome to a special edition of Going Antiviral podcast, recorded live from the 2023 Ryan White Clinical Conference in Portland, Oregon. Today, we're very fortunate to be speaking with Dr. Richard Koup, who's Deputy Director Chief, Immunology Laboratory Vaccine Research Center at the NIAID NIH. Dr. Koup, thanks for joining us.
My pleasure.
You've been at NIAID now for 22 years. And through that time, you've witnessed a lot. You've been in the heart of vaccine development, not only for HIV, but also other vaccines, most notably COVID. We're going to talk about that in a little bit. But I'd like to take us back to 1983 or 84 when HIV was discovered in the Gallo lab and also in France. And Margaret Heckler, the HHS secretary at that moment, said, "We're going to have a vaccine in 2 years." What happened?
Well, thank you, Dr. Saag. Yes, 1983, everyone remembers that quote from Margaret Heckler. In fact, what people don't realize is she didn't say we'd have a vaccine in 2 years. She said we'd have a vaccine in clinical testing in 2 years. So she got a lot of flack for saying that because we had never developed a vaccine in 2 years. And in fact, if you look at the historical record, it was 1987 when we had the first vaccine candidate in clinical testing. So it was 4 years. So she wasn't that far off.
Well, thank you for defending her honor. I think we get that wrong. I certainly got that wrong. But you mentioned, I'm going to make note of that 2 years developing a new vaccine a little bit later in the conversation. But let's focus on HIV to begin with. Take us into a little bit of the basic science. Let's start with the virus in terms of trying to describe why it's been 40 years and we still don't really have the vaccine. So looking at the virus, what's the nature of it that makes this so challenging?
Yeah, it's a very good question. So I like to think of this as a problem of the 40s. It's been 40 years. We continue to have about 40 million people living with AIDS or HIV, and still we don't have an efficacious vaccine. We've spent about 36 years testing vaccines. We've spent 20 years actually doing efficacy trials, vaccines that we thought would actually protect, and still nothing. Why? Well, you mentioned Bob Gallo very early on. He basically, nailed it very early. He said, this is not going to be an easy virus to protect against because you probably need to get sterilizing immunity. This is a lentivirus, which means that it actually replicates itself, inserts a part of its genome, actually its entire genome, into the cells of the body. And then that genome remains dormant. Think of it as like the groundhog. So it goes dormant for part of the year, and then whenever it wants to, it can just reemerge on Groundhog Day or whenever. And as a result of that, a standard vaccine, what you do is you don't protect against infection, you protect against disease and severe symptoms. So with the COVID vaccine, which I know we'll talk about later, most of the vaccine efficacy doesn't block all infection. You still get infected, but your symptoms are much less. With HIV, you can't do that. You have to block all infection because otherwise some of that virus is going to get into your cells and it's going to be there forever and it can come back at any time.
That's a great point. Some of the work I did early on when George Shaw and Beatrice Hahn moved from Gallo's lab in 1985 to UAB, where I was a fellow and we worked in the lab. And one of the things we focused on was the genetic variation of HIV. It's one of the first studies I ever did. And what we found was that within every person, there's at least 15 to 20 detectable circulating viruses that are genetically related but very distinct than so-called the quasi-species. So does that challenge you as well, not just being a groundhog, but a groundhog of different stripes?
Absolutely. So, most viruses, when we try and develop a vaccine, we're talking about 1, 2, maybe 3 different strains that we need to cover. Influenza, as you know, is much more difficult because it has this drift and this shift. So you have many more different forms of the virus that you need to protect against. HIV is a magnitude, an order of magnitude worse, in that you have so many different viruses that replicate within an individual. And then if you look out in the population, the number of viruses out there and their immunologic differences is huge. So your vaccine needs to cover all of those. And it's very, very difficult to get a vaccine that covers, even one, much less all of them.
So one of the ways that vaccines could cover to create the sterilizing immunity, I suspect, would be to get antibody. That we know that there's an antibody against one of these genetically distinct viruses, but 2 weeks later, a new variant could pop up in that person and that antibody has no activity. So we're really talking about this concept of broadly neutralizing antibodies. Can you describe what those are? It's often called BNABs.
Yeah. So BNABs, or broadly neutralizing antibodies, are antibodies that develop within people that can neutralize many, many, many of these strains that are out there in the world. We have broadly neutralizing antibodies that can cover 96, 97, 98% of strains. So these really tell us that if the immune response is correctly focused, that actually you can generate immunity that will cover most of the viruses that are out there. Now, generating these types of broadly neutralizing antibodies through vaccination is very difficult, and is something that is really one of the targets of our vaccine development at this point.
So to sum it up, the idea, since we know that there's all kinds of different groundhogs that hide out but or look different from one another, we want something that will cover all of these or as many of these as possible through broadly neutralizing antibodies. And if we can get a vaccine that stimulates, let's say, coverage of 95%, that should translate into a degree of effectiveness in the population. What would you say would be success? Would it be 30% protection of new infections, 50%? We'd, of course, love 100%, but I don't know of any vaccine that does that. What do you think?
Yeah, obviously, as you say, we would love to see what we saw for the initial COVID vaccines of 95%. I think if we got over 50 or 60%, that would be a good starting point. And certainly a vaccine that showed that type of efficacy is one that would have a major impact on the epidemic. I think that, however, would not be the final vaccine. That if we had that kind of efficacy, we would keep striving. We would learn from that vaccine. We would make improvements and go for better and better immunity.
And one of the advantages, just putting it all together again, is that unlike influenza, you would have a vaccine that could cover against drift and strains emerging. And getting 60% efficacy would definitely bend the epidemiologic curve. It wouldn't stop, but you put together PrEP and treatment as prevention. This could be the sort of final holy grail that could lead us realistically towards stopping HIV transmission in the community.
Yes, you're absolutely right.
So that's the idea. Okay, let's dig a second into how we learned about BNABs. If I remember correctly, the first time I heard about it was Bart Haynes out of North Carolina, Duke, who was looking at some lupus patients, if I remember correctly. You can get me straight on the story, but basically noticed that very few lupus patients were getting infected with HIV, and he started looking at antibodies, and we know that people with lupus produce a lot of antibodies. Is that where it came from, or was there other sources?
I think, as is typical with most scientific discoveries, there wasn't just one. And actually, the story I know best is the one that came out of the Vaccine Research Center, where within the NIAID, the National Institutes of Allergy and Infectious Disease, there was a guy called Mark Connors who was following these large cohorts and found that some individuals, if he looked at their serum, they had very good neutralizing activity and others didn't have good neutralizing activity. And so it was actually technology out of Michelle Nussensweig's lab from the Rockefeller University that allowed us to then sort B cells out of these individuals who had broad neutralizing activity in their serum and amplify the immunoglobulin genes out of those B cells. And then we could screen those immunoglobulins and see if they neutralize broadly. And that's where the first broadly neutralizing antibody called VRC01 was discovered.
So how many patients would you guess out of the universe of folks who are infected with HIV have plasma cells or B cells that produce BNAbs?
Yeah, that's a good question, and it's often said, oh, about 10% or 20%, but it's actually a continuum. And Betty Korber points this out. I mean, if you look, it's just a continuum, and it all depends upon where you set your definition of broadly neutralizing. And so wherever you want to set it, you can say 30%, you can say 40%, you can say 10% have really broadly neutralizing antibodies.
The punchline then is that you study the cells that produce these broadly neutralizing antibodies from a number of different people who are producing them as part of their natural HIV infection, and then you try to study sort of the mechanism of how those plasma cells that produce antibody evolve. Where do they come from? How can you make it happen in everyone who's never been infected before? That's kind of the pathway to getting a vaccine that does that. Is that right?
That's absolutely right. So once we have a broadly neutralizing antibody, we can identify what part of the envelope glycoprotein, the HIV GP160, that it binds to. Once we know that, we can go back into that individual that we isolated the broadly neutralizing antibody from, and we can do sequencing of the B cells over time, and we can say, here's how we can see that antibody, what it looked like initially, then how it continued to change over time to get to be broadly neutralizing. And what we find is that the first neutralizing antibody will be very focused. And it'll be focused on the envelope of the HIV that the person that it came from had at that time. That envelope will then escape that response. And through that change, it forces the antibodies to change, which force the envelope to change, et cetera, et cetera. And so it takes a long time of the envelope changing to get the antibody to change and mature and become broadly neutralizing.
Do you think you can shortcut that? In other words, by giving a vaccine, not require somebody to go through a years of cat and mouse with virus versus antibodies? That's the challenge, right?
That is the challenge, and it may be very difficult to just start at the end with the antigen that the person had, or the envelope that the person had when they had the broadly neutralizing antibody. I think, and most of the strategies right now are you start at the beginning. You try and get that initial response from the immune system. Once you have that, then you start changing the antigens that you're going to, or the immunogens that you're going to give to an individual, such that you will mature the response into a broadly neutralizing response. What that means is that you're not going to get the same vaccine every time you go to the doctor. You have to get vaccine number 1. And then a few months later, you go back in for vaccine number 2. And a few months later, vaccine number 3. And it's only in that way that you'll mature the immune response to get the kind of antibody you need. That's going to be complicated, unfortunately.
So we focus this entire time on neutralizing antibodies and sort of the humoral side of the immune system. Is there any continuing work on the cellular side where you prevent viral infection by stimulating a person's immune system to recognize a virus and then get its CD8 cells or whatever to attack it, prevent it from ever infecting?
Yeah.
What's going on there?
Yeah, that's a good question. A lot of the vaccines that have failed so far have induced those types of classical CD8 T cells that you mentioned. There is one group that has worked on a vaccine with a viral vector that induces very strange, let me call them very strange CD8 T cells. They don't recognize the virus the way CTL usually recognize virus, but they recognize it in a way that's very atypical. Interestingly, in studies done in monkeys, it's been shown that that vaccine is protective at 55% efficacy. And that's sort of right on that border where I said if we had something that worked at that 50 to 60% efficacy, that we would be in good shape. And so that vaccine is currently in clinical testing.
That's pretty exciting. I didn't know about that. So that's great. Well, we've spent some time on HIV, and I'd like to sort of give my tip of the hat to Margaret Heckler and take away my sounding critical of her. But we have learned a lot in 40 years, even though we don't have a vaccine in hand. And I think what it underscores is how difficult it is to develop vaccines against new pathogens. And with that as an intro, COVID. I mean, I remember when I want to hear the story on the inside that you can tell, but I remember when I heard about the efforts in January, February of 2020 that they're working on a vaccine. I'm thinking, yeah, that'll be for 10 years from now, because that's about what it takes for all the reasons we just went over. But yet, within 11 months, we had not only an effective vaccine that produced 90% efficacy, which we talked about 40, 50, 60%, we'd be happy, 90% efficacy, and that it could be mass produced and delivered by, I think I got my injection on December 20th of 2020, the same year that you started. So let's go back to January 2020. You're at the National Vaccine Research Center at NIH. What happened?
So it was really a convergence of many things. So as you're aware, Moderna was one of the companies that came out with the vaccine very quickly. The Vaccine Research Center had actually been working with Moderna to try and to see how quickly vaccines could be developed. They had this mRNA technology, and we felt maybe this could very quickly get us from discovery into vaccine testing, because you don't have to make proteins, purify proteins, et cetera. You can just take the mRNA, and it's much quicker to get into people. As part of that, we already had the infrastructure of dealing with Moderna. In addition, we had many years of research into similar viruses to SARS-CoV-2. We had worked on MERS. We had worked on SARS-CoV-1. And so we knew how to stabilize the spike protein of those viruses. So when SARS-CoV-2 came out and we saw the sequence, it was a matter of less than a week, and the mRNA was being produced to make that vaccine at Moderna. So it was because everything was in place with the right agreements, everyone working together, that we were able to get it into testing. And then mRNA, the whole was very quick to move forward. The U.S. government got involved. People from all walks of life started helping and working together, and it's why we were able to move so quickly.
I mean, I wrote a paper, just a summary of not exactly all of that, but just the development of a vaccine and all the response to COVID. That the title of it was Wonder of Wonder, Miracle of Miracles, because it really feels like a miracle. But I think what it's underscoring is something that the general public perhaps doesn't fully understand, and that is the value of basic science research. Unpacking your comment, years of work on MERS and SARS-1 and Ebola to some degree, and other vaccines, mRNA technology coming along. When was that? About early 2000 or so, and Drew and others were sort of working on Drew Weissman.
Well, Drew Weissman and all had been working on it for many years, but yeah, the taking up of mRNA technology into vaccines, was
a decade.
Yeah.
So all that work that seemingly might not go anywhere is sitting there for the picking, and it comes together, converging is a great word, into this remarkable, astounding advance that I think even still the general public doesn't quite understand the miracle of that. But I think when you put it in the context of an HIV vaccine and how hard you and your team and all of the National Vaccine Research Center and other investigators have been slaving away at, shows in contrast what a miracle this COVID vaccine is and was.
And thank you for saying that, but I really should say that when I said we, I wasn't talking about me or my team. I should really give a shout out to Barney Graham, Kizzy Corbett, and Jason McClellan, who were the two people at the Vaccine Research Center and a former VRC employee who really were the drivers behind this.
So the vaccine mRNA starts being produced in January into February, and then it rolls out into clinical trials. And that's also a quasi-miracle, because just let me review and then you tell me what happened. What normally happens is you have to test it in animals and make sure it does what you think it's going to do. And then you've got to take a candidate to see if it's safe in people and whether it does what you thought it was going to do. That's phase 1, 2. And then when you see that, you got to find the right dose. And then you take that into phase 2B and ultimately into a population phase 3 to look at its efficacy versus a placebo-injected group. And normally that process that I just easily spouted out is a 5-year exercise with regulatory oversight by FDA and other sources and funding that has to come from somewhere to support the work. And all this was done in a matter of months. Oh, and I forgot protocol development, which people usually argue over for 6 months. How in the world did that happen, Rick?
It was basically that people realized that this was a problem and decided to come together. So Operation Warp Speed, the U.S. government brought, the FDA, the CDC, BARDA, NIH together and said, we need to fund companies. They gave money to 6 different companies to work on vaccines. And we had a huge group of both academic and government officials overseeing that process and forcing it forward, so that there is a certain way of doing things within industry to reduce risks. You don't want to spend a lot of time and money doing things where the product may fail and you lose all that investment. Well, the fact was the government invested that money. And basically that allowed the companies, and we forced the companies to take risks that they normally wouldn't. And I'm not talking about risks to the patients. The patients were not the ones that were put at risk. It was the investment in money that was
So it was a business risk.
Yeah, it was a business risk, not an individual human risk.
Yeah. So the other thing that is hidden in your comment right there is that once you invest whatever it's going to be, I'm going to put a number on it, 300 million to 500 million to get through phase 2 protocols, and you have success, then you've got to scale up, right? We got this, yay, we won, we got a vaccine, but then you got to mass produce it. And that takes plants and regulatory oversight and good manufacturing processes and all that. That was happening in parallel.
And that's part of the risk mitigation. So yes, a company isn't going to do that till they know that the vaccine works. We said, even before the vaccine works, we're giving you the money to start producing lots of the vaccine. So such that if it works, we've got it. If it doesn't work, the government has lost that money, but not the company.
So getting this straight, while they're still in phase 1, 2 clinical studies, over on the other side, they're building plans to produce something. They don't have a clue if it's going to be effective.
Correct.
That's amazing. So finally, we get to November of 2020, and the studies start to have their results come in. Tell me your personal story of when you first saw the data.
Yeah, I mean, we all thought, well, it would be nice if we saw some efficacy. When we sat down with the different companies and saw those data, it was overwhelming. I mean, you look at the Kaplan-Meier curves, the risk curves versus showing the placebo versus the vaccine recipients, and after about 1 week, 2 weeks, those curves start to split. It was unbelievable. I mean, in the HIV field, when we're looking for efficacy, we're trying to say, oh, is there 30, is there 40% efficacy? This was just unbelievable. I mean, there was just no question. And I know Barney Graham, who was very involved in the development of these vaccines, he just lost it. I mean, it was just, it was emotionally. I mean, it was just very hard for him to believe just how fantastic these vaccines worked.
It's one of those moments like, where were you on 9/11? If you were alive at the time, where were you on Kennedy got assassinated? That's a big moment.
Yeah.
I mean, I had something similar with when viral load, we stumbled onto that as quantifying it and saw it just on people who were asymptomatic, had low level, and people who were AIDS had high. And it's the way I describe it is, you don't need a statistician to see that there's an impact here.
Yeah.
Well, so when are we going to get to that with HIV?
Yeah. Well, as I mentioned, HIV is a very difficult virus, even though we've got all the basic science that you said was so important. Now that we've got mRNA, and that mRNA allowed us to move much more quickly with SARS-CoV-2. SARS-CoV-2 and HIV are completely different viruses. We've got to get sterilizing immunity to HIV. And so that's where the difficulty is going to be. So it's still going to be many years of a hard slog, but we're optimistic that we've got something now. We've got a pathway to get there.
Well, this has been a wonderful podcast. We've gone through the challenges of HIV, how it's a different virus. It has multiple different forms of itself, different types of groundhogs that hide in cells that can come out whenever the immune system has to respond. We got to find ways to cover the waterfront with ideally broadly neutralizing antibodies, and maybe combine with some cellular immune cells with these strange CD8 cells that you're talking about. Maybe we can get that together. We can bend the curve if we add that type of prevention technology to what we're doing with PrEP and what we're doing with treatment as prevention and what we're doing with testing and treating. And maybe there is hope for the future here, and we're going to find ourselves in a good position. Let's finish up with your projection of where do you think we're going to be 10 years from now?
Oh, I hate it when people ask things like that, because we should never try and predict the future. We should never try and say where we're going to be in 10 years, because we're almost certainly going to be wrong. But I would certainly hope that we're much further along on that pathway. And either we have something that's looking like it's going to work, or if it hasn't worked, we know why, and we've learned from that process.
It's like George Harrison said, and while my guitar gently weeps, with every mistake, we must surely be learning.
Yeah.
And I think we are. Dr. Koup, thank you so much for being with us on this podcast, Antiviral. We are finding ways to use technology and basic science to make our world better, despite what some naysayers and haters out there would say. Thanks for setting the record straight for us and being with us.
My pleasure. Thank you, Dr. Saag.
Thank you for listening to this episode of Going Antiviral. This podcast is for informational and educational purposes only and is not intended to serve as medical advice. Opinions expressed in this podcast do not necessarily reflect the opinions of the IAS-USA or its affiliates. To listen to more episodes of Going Antiviral, be sure to visit our website at IAS-USA.org.